The human skin is the largest organ of the body, but despite forming a single, continuous covering, its architectural construction is far from uniform. The structural requirements of the integument vary wildly by anatomical location. This is evident when examining physical extremes, like areas requiring the least mechanical armor. This text highlights regions where the skin is at its thinnest. Over the eyelids and penis, total thickness measures 0.5 millimeters. Contrast that with regions demanding mechanical resilience. In the sole, palm, and interscapular region, the skin scales up to 5 millimeters thick. Between these two extremes, the average baseline thickness across the rest of the human body settles at about 1 to 2 millimeters. How does a single, contiguous organ system execute and maintain such massive localized discrepancies in its physical depth?
This structural variance relies on a bipartite architectural model, two distinct, deeply interdependent layers working as a single unit. This bipartite system divides the skin into an outer layer, the epidermis, and an inner foundation, the dermis. The epidermis serves as the primary environmental barrier, structurally classified as a stratified epithelium. As noted in the documentation, the defining physiological constraint of the epidermis is that it completely lacks blood vessels. Because this outer layer is avascular, it extracts 100% of its nutrition through diffusion, pulling resources upward from capillary networks housed in the dermis below. This avascular layer dictates the design of the tissue beneath it. The outer barrier's survival relies entirely on the complex architecture of its deep foundation. To understand this support mechanism, we must look closely at that inner foundation, the dermis. The source text defines the dermis as a connective tissue layer, primarily composed of dense, stout collagen fibers, alongside elastic fibers, fibroblasts, and histiocytes. These collagen fibers exhibit highly specific physiological capabilities. They possess elastic properties capable of retaining water, and they contain collagenase, an enzyme explicitly responsible for wound healing. In a 3D dermal cross-section, the deepest zone is the reticular layer's chaotic fiber mesh. Above sits the superficial papillary layer, introducing chromatophores and nerves. Upward projecting dermal papillae tightly interlock with downward rete ridges. Inside each papilla, perpendicular capillary loops solve the avascular paradox. By pushing these vertical, blood-rich projections deep into the epidermal boundary, the dermis drastically increases the total surface area available to diffuse nutrients to the layers above. With the life support infrastructure in place, we track the cellular journey upward into the epidermis. The foundational layer of the epidermis, resting directly on those papillae, is defined here as the stratum germinatavum. Structurally, this germinatative zone consists of a thick, active bed of polygonal cells. Moving one step toward the surface, we arrive at the stratum spinosum, frequently designated as the prickle cell layer. Here, cells develop distinct, spine-like protoplasmic projections extending outward from their membranes. These projections serve a vital mechanical purpose. They physically connect adjacent cells to one another, providing the shear strength required to withstand external tension. These deep, living basal layers provide both the cellular raw material and the robust structural anchoring required to construct the environmental barrier above. As the newly generated cells migrate higher, they enter the stratum granulosum, compressing into a thin layer of 2 to 5 rows of flattened rhomboid cells. At this depth, cytoplasm accumulates dense deposits of keratohyalin granules, the chemical precursor to keratin. Pushed upward into the stratum lucidum, cells exhibit a shiny, translucent character. Here, cellular morphology undergoes severe degradation. Driven by physical pressure, the cells rapidly degenerate, completely losing their nuclei. Finally, this migrating cellular material reaches the surface boundary, the stratum corneum. The cells in this final layer are known as corneocytes. They are entirely dead, their cytoplasm thoroughly flattened, and packed with mature fibrous keratin. The epidermis operates as an assembly line of programmed cellular death. It systematically transforms living polygonal cells into a resilient, continuously shutting armor, composed of keratin, phospholipids, and glycogen. This layered block of tissue is further integrated by specialized structures that transverse the strata, collectively known as appendages of the skin.
These primary appendages include nails, sweat glands, sebaceous glands, mammary glands, and hair follicles. To function, a hair follicle must bridge the organ's internal divide. It is driven down through the hardened layers of the epidermis, anchoring securely deep into the reticular dermis. This deep anchoring allows for the integration of the erector pili, bundles of smooth muscle embedded directly within the skin tissue. These smooth muscles are positioned strictly adjacent to the follicles, attaching directly to the side of the submerged hair root. These appendages integrate the integumentary system into a deep, interactive three-dimensional matrix. Looking at the organ macroscopically, the color of the skin depends on two primary factors: the volume of hemoglobin in the underlying blood and the concentration of cellular pigmentation. Examining this outline on pigmentation, we see that skin color is driven by specialized cells called melanocytes. These melanocytes are stationed precisely within the living basal layers we mapped earlier, resting primarily in the stratum germinativum and the stratum spinosum. Deep within these layers, the melanocytes continuously synthesize a protective brown pigment known as melanin. But once synthesized, this pigment does not remain isolated in the lower strata. The melanin actively spreads outward from the melanocytes, permeating the surrounding cellular structures and migrating upward into the higher layers of the epidermis. A localized chemical process at the deepest boundary of the epidermis diffuses upward, providing a global protective feature across the entire surface. The skin operates as an integrated biomechanical system where the outer avascular barrier is sustained, armored, and pigmented by the deep vascularized architecture of the dermis below.